Hello everyone, this is Hunter Francis again, joining you from Cal Poly State University in San Luis Obispo, California. Welcome back to our online educational series on soil health, funded in part by the USDA Western SARE program. Today we're going to be talking about organic matter, normally a relatively small part of the overall soil makeup, but a very important one, especially when it comes to biology. The organic matter in soil essentially comes from anything that was once living, residues of any type of plant and animal material that has made its way into the soil and is at various stages of decomposition. Organic matter is usually something that differentiates soil from mere unconsolidated mineral or rock parent material, such as beach sand or alluvial deposits. Over time, organic matter builds up and makes its way down into the soil profile, changing the soil's color and a lot of its inherent capacities along the way. There are such things as quote-unquote organic soils, often part of the soil order called histosols, and those usually come from bogs or wetland areas that have dried out. They can contain an abnormal amount of organic matter, but in most cases, particularly in agricultural soils that have been under cultivation, organic matter is only usually around 5%, and sometimes much less in drier areas such as California, where it's normal to find organic matter at only about 1%. In a good agricultural soil, only about half of the makeup from the mineral soil particles as indicated in this pie chart, leaving another half as pore space that is ideally filled with about half air and half water. Of the organic matter fraction, at least 3/4 of it is typically comprised of stable organic matter, traditionally called humus. The other quarter is comprised of organisms that are still alive, or more freshly deposited materials that are more easily decomposed. It is common nowadays for soil folks to talk about three basic fractions of organic matter as the living, the dead, and the very dead portions. Boundaries are somewhat fluid as the organic portion of the soil is dynamic, and site conditions also play into variability, which is why this pie chart is a bit different from the previous. But again, the living organisms in the soil make up a fairly small portion of the organic matter, and normally only about 10 to 15% of the dead material is readily decomposable and available to be mineralized into plant nutrients. Of all the materials combined, about half is comprised of carbon, or between 50 to 60%. About 3 to 7% of the organic matter is comprised of nitrogen, which can be more or less available, in other words, more or less readily mineralizable. The plant and animal residues, secretions, and byproducts that are found in soil are quite varied. As you might expect, the materials found are the building blocks of life, such as carbohydrates, fats, and proteins in the case of plants, things like cellulose and lignin. As mentioned in the last chapter, chemical metabolites such as enzymes can be very important in the functioning of and communication within the soil food web. As the fundamental building block of living tissues, carbon finds its home in organic materials of all sorts and is in a state of flux and interchange between such materials and carbon in its mineral form in the atmosphere as carbon dioxide. This figure is a quantification of the amounts of carbon in the different spheres of the Earth's ecosystem. And it's interesting to notice that the carbon in the soil is over double all of the carbon that's contained above ground in the biomass of plants and animals, plus all of the carbon contained in the atmosphere above them. In this case, if one looks to the left and discounts where carbon exists in the oceans and sediments, soil organic matter consists of over 1.5 metric gigatons of material, or about 1.8 billion US tons of materials, and this seems to be a low estimate. What this means is that when one looks across the landscape, even in a forest with all of its abundant vegetation, one can expect that there's well over twice as much carbon contained in the soil beneath it. The gateway to much of that underground carbon is plants. It's true that the soil is fed by the residues of decaying plant and animal material, but a significant portion of the carbon that makes its way to the soil comes through plant exudates, and then makes its way down into the profile through what is sometimes called the liquid carbon pathway. This withdrawing of carbon from the atmosphere through photosynthesis is called sequestration, a kind of trapping of carbon, and it is receiving a lot of attention these days because of the desire to get carbon out of the atmosphere. The concept of drawdown is a strategy for atmospheric carbon reduction that more and more planners and engineers are considering to supplement carbon emission reductions, and it's made possible by plants. What is illustrated in this schematic is the fact that this drawdown sequestration scenario can be a positive feedback loop, and it underlies many of the strategies for agriculture to build healthy soil, because by introducing more plants into the system, keeping soil covered, and promoting the diversity of life forms on top of the soil, one builds carbon in the soil, which in turn feeds the microbiome. This consequently helps to promote better and more vigorous plant growth and allows for more drawdown of atmospheric carbon. This is an important dynamic, and we can expect that it will be receiving more attention. The nice thing about it for agriculture is that it has the potential to be a win-win scenario between farming and society at large. A little blast from the past here. I'm afraid I have a fascination with history, and I guess what fascinates me about it is how ideas flow and morph through time. A lot of times new ideas really aren't so new. They often start small, perhaps evolve a bit, and take time to gain recognition. Albert Howard is often considered one of the founders of the organic agricultural movement. As some of my colleagues observe, some of his ideas are outdated, but he did develop them almost a century ago, long before he wrote them down in his book, An Agricultural Testament, in 1940. I mention him here because we've been talking about cycles of carbon, and a central point that Howard eloquently expounded upon was the idea of the law of return, which refers to the fact that nature returns everything that it uses back to the earth. The idea was that we should try to mimic that process in our agricultural systems. The idea of feeding the soil was a basic tenet of early movements in the alternative ag sector and is now finding wider acceptance. When you think about it, the process of use, return, and reuse, recycling, closing the loop, and so on, is at the core of many sustainability strategies and is something that transcends both conventional and organic. It makes sense in many systems. When organic matter is cycling successfully and finding residence in the soil, the benefits are many. Tilth is improved, bulk density lessened, aggregation of soil particles increases and they become stronger, creating more permeability and infiltration of water and less erosion. Organic matter also increases the buffering capacity of soils, softening extremes in things like pH and temperature, and helps with nutrients as well as water. I would like to touch a little bit more on some of these benefits in the next few slides. As we've said, organic matter plays a vital role in aggregation that occurs in soil. It produces cementing agents, as they're often called, the glues and sticky substances exuded from plants, and plant roots and hyphae themselves can act as kinds of wires or scaffolding, holding the various particles in place. The tiniest of particles, called colloids, which are microscopic, have a tremendous amount of surface area, and they contain charges that can help hold ions to them. Fundamentally, there are two types of colloids that possess charge, mineral and organic. This schematic shows the charges around the mineral clay particles that are fundamental in holding many of the elements beneficial to plant growth. In neutral to basic soils, the majority of these charges are negative, which means positively charged cations are attracted to the particles. These ions attach and detach to the colloidal surfaces in a type of exchange. The amount of ions that a particle can hold is its capacity, and when the charges are negative, they have the capacity to exchange cations, positively charged ions. This is the cation exchange capacity of soils that is largely contributed to by clays and organic matter. Nutrients flow through the soil solution between plant roots and the small soil particles as a part of this exchange. The attraction to the soil particles is more of an electrostatic attraction as opposed to a permanent bond. Thus, the particles are able to hang on to the nutrients and then release them when needed by the plant. This exchange of cations often includes the release of positively charged hydrogen ions from plant roots, especially when plants take in negatively charged nitrate, which is one reason why over time in older and highly weathered soils there's a tendency towards acidity as cations are consumed and leached and hydrogen builds up on the cation exchange sites. The chemistry involved in cation exchange is somewhat complex, but we've listed a few basic considerations here. As has been stated, it is the negative charges on soil particles that create the capacity to attract positively charged cations. However, that charge on the particles can itself fluctuate, largely due to pH. Particularly on organic matter, negative charge increases as pH increases. There are, however, some negatively charged sites, particularly on clay materials, that do not change with pH and are thus considered permanent. In the measurement of cation exchange, depending on the laboratory technique used, CEC may reflect either total cation exchange capacity or what is called exchangeable cation exchange capacity. The latter refers to the ease with which cations are released from the exchange sites. Exchangeable cation capacity is determined in the lab by mimicking conditions in a normal soil solution and by pulling off and measuring what ions would be readily removable and available for plant nutrition. However, there are some cations which are more tightly bound, thus adding additional cation exchange to the total exchangeable ions, some of which are not easily released in normal conditions. Part of this has to do with the nature of the actual ions that are on exchange sites. Depending on the charge of the element and also its hydrated radius, which basically has to do with the sphere of its relative charge and can be either large or small. Certain elements with multiple charges listed here to the left, like aluminum and calcium, are held tighter to exchange sites than elements with a single charge, like ammonium and sodium. CEC is measured by what are called milliequivalents of charge per 100 grams of soil. Typically, the CEC will range between 0 to 50 milliequivalents of charge in most soils, with sands being on the lower end and clays being on the higher end of exchangeable CEC. The term pH actually comes from the French word pouvoir, which means power, hydrogène, which is hydrogen. pH refers to the power or the strength of the concentration of hydrogen in a solution. It's actually a negative logarithmic number, so as pH increases, and because pH refers to a negative value, there's less hydrogen in the system. A lower pH of, say, 3 or 4 means that there's more hydrogen present. In a lower pH system, positively charged hydrogen ions will bond and fill up the exchange sites, as on the left here, thus making those sites unavailable for cations and therefore decreasing the exchange capacity as shown in this picture. I'd like to turn back now to the fundamental nature of organic matter in terms of the degradability of its substances. We talked about the living, the dead, and the very dead, but a more traditional way of talking about organic matter is a dichotomy between what's called labile organic matter or stable organic matter, which has traditionally been understood as humus. On the labile side of the spectrum, you have biological organisms themselves, such as bacteria and fungi, whose membranes are easily decomposed when they die, thus spilling their contents out into the soil with easily digestible compounds such as sugars and fats. Those and freshly decomposed detritus and other particulate organic matter is more or less consumable by other organisms. Particulate matter that is protected and physically hard to access by those organisms, or compounds that have somehow been transformed, perhaps through fire or geological processes, as in biochar or coal, are forms of carbon that are much harder for the microbiome to use, thus making them stable in comparison. The types of detritus or plant or animal materials deposited into the soil in terms of their molecular complexity can be a factor in terms of this access. This graphic showing the chemical formula for a simple sugar like glucose as compared to one that's more complex like cellulose, or even further complex like lignin, which is highly complex and does relate to degradability because it's harder for microbes to break apart the bonds of larger molecules. Thus they are less degradable and are what is called recalcitrant, that is, not prone to decomposition. Chipping away at some of these larger molecules is one of the roles of organisms that are of a higher order than bacteria, such as fungi and earthworms, because through their digestive processes they help decrease the complexity of some of these molecules and make them more available to the little guys. Ideally, we would like carbon to stay in the soil and not all eaten up and gassed off as carbon dioxide as microbes respire. Thus, we want a good portion of our carbon to be on the stable side of the spectrum. For a long time, that complex, stable organic matter was called humus, not to be confused with humus, and has been prized by agriculturalists throughout time, a holy grail, so to speak. This is a molecular representation of a humic substance, and like the lignin that we just saw, it is even more complex and made very stable by the many aromatic or six-sided rings that you see in the diagram. Humic acid and its cousins have hydrogen wings, so to speak, or protonated tentacles that contain hydrogen and can be released to form salts, in this case, humates such as calcium humate. Humic substances, such as humic acid, are commonly found as agricultural inputs, and they're often mined from old deposits of organic matter. Humic chemistry is very complex and is related to coal chemistry, and a large range of compounds that have carbon at their core are highly complex and stable. The basic fractions of humus are depicted in this flow chart and are humic acid, and fulvic acid. These fractions are separated through a highly extractive and really a very unnatural laboratory process which exposes a soil sample to a very strong base, sodium hydroxide in the upper left. That which is not able to go into solution and then remains in a solid form as a residue is called humin. From the extracted fluid, a very strong acid solution is introduced, at which point humic acid precipitates out and the fulvic acid remains in solution. Simply speaking, humic acid is a longer chain, more complex molecule, whereas fulvic acid is a shorter chain humic substance and is able to remain solvent. This can make it more easy to use in things like drip lines than humic acid. Revisiting the charge that is related to cation exchange capacity, which we just spoke of, these are the types of chemical groups found at the periphery of humic substances. Those OH groups, or hydroxide groups, that are part of the carboxyl, phenolic, and hydroxyl appendages are what help give organic matter its charge, because the hydrogen can be released, and when they leave, particularly in a basic solution, a negative charge results, as seen on the right of this picture, thus enabling the molecule to attract and hold on to positively charged cations like calcium and magnesium. This understanding of humus, which we've been speaking about, has been with us for a long time. In fact, the study of humus dates back several centuries to the late 1700s when it was first discovered. The chemistry, in particular the molecular structure and size of the compounds, which we have been talking about, has been understood to be the primary factor as to why these humic substances are so stable. This chart shows the flux between organic residues at the top, going through the quote-unquote eye of the needle of the microbial world, through all the degradation processes that happen underground, resulting in more complex humic substances. This was the basic understanding of how humus was built up, and which I was taught. It was through both the microbial degradation, but then also the synthesis of more complex molecules that made that portion of the organic matter so stable. Recently, however, this whole understanding of humus and how it is built up and preserved in the soil has been called into question, notably by a paper in 2015 by Lehman and Kleber. That, along with other research, has generated a huge discussion and, to some degree, some controversy within the soil science world. The basic premise here is that it is less the molecular structure of the molecules, including humic-type molecules, that make them stable, as opposed to other factors that render them more or less available to microbes for further degradation. As you see here from the top to bottom, the residues are going through a similar breakdown cycle, but the notable difference is that on either side you see complicating factors, whereby some of these molecules are either transformed and held into aggregates, which can be quite tight and, when working well, strong, protecting and creating a kind of physical barrier to the organic matter, or on the other hand by absorption onto mineral surfaces. That likewise makes them difficult to pull apart. The nuances between the two models are fairly sophisticated, but I'm mentioning them here now because it's very clear that the understanding of humus is evolving, and that we will be talking more about it and discovering new things about it as science advances. Nonetheless, and yet this is another schematic or another way of comparing organic matter, for a variety of reasons, the organic matter in the soil will be more or less active or passive, with the active or more labile fractions, which are a relatively small proportion of the pool, being more readily decomposable, and the passive remaining longer in the soil. The CN ratio of the compounds can play a role in this, with lower CN ratios being more easily degraded, and we will talk more about this in the next chapter. What is being posited by new theories of the relative stability of organic matter that adds to our understanding of humus is the idea of protection. This somewhat complex schematic is merely showing, in another format, that in the flux between chunks of organic matter on the left, their assimilable form in the middle, and their use by microbes on the right, some of this organic matter will be attracted to and bound with soil particles, or be trapped or occluded within them, making them inaccessible, at least for the time being, to degradation. Added to this, environmental factors such as cold temperatures or the lack or excess of water, of course, can also be an impediment to degradation. Another way of looking at this that's of concern when we're trying to achieve a net buildup of carbon in the soil is the polarity between the release and the capture or holding on of carbon within the system. On the one hand, you have the idea of priming on the left, which refers to the fact that carbon, particularly labile carbon, is a form of food for microbes, and when given to them, will make their populations expand. Therefore, more organisms and more grazing, so to speak, of soil carbon, which means more loss of carbon from the system through respiration. On the other hand, and what we want, is a buildup of the carbon in the form of stable organic matter. And when this is achieved, you have what's called entombment of organic matter. And this is largely assisted by the physical and chemical protection mechanisms that we just discussed. What we should remember, however, is that even in a priming situation where you're feeding respiring microbes, you're also building the biomass, the actual carbon contained in the organisms, which then becomes what is called necromass, or dead biomass bodies. And that results in carbon that does have the potential to become entombed as stable soil carbon. And what is recently drawing scientists' attention is the fact that there may be a difference between organic matter carbon of plant origin versus microbial origin, with findings seeming to show that the carbon of microbial origin may be, in fact, more stable and less prone to degradation after it has passed through the microbial eye of the needle. Thus, it is thought that the microbiome creates a kind of pump that is introducing a more stable form of carbon into the system for long-term benefit. The microbial pump concept is rather new, but if it holds up, it could be one way of better understanding the nature of organic matter molecules and how that understanding fits into new paradigms. The moral of the story is we need to build our carbon. In other words, build up organic matter. We will be looking at a new framework for tracking, evaluating, and planning for carbon in soil in the later part of this series. But for now, we would just like to point out that the concept of carbon farming is starting to gain traction, particularly here in the West. It basically involves an assessment of what carbon exists and how it is flowing on a particular farm or parcel, and how that carbon might be augmented and held better through soil building practices, planning for implementation of such practices, and inventorying what incentives, particularly NRCS soil conservation incentives, that might be available to help underwrite the adoption of such practices. You can find out more about carbon farming from the Carbon Cycle Institute website, which is listed on this webpage. In a world focused on NPK, carbon is often a forgotten nutrient, but as we've seen, it can be one of the most important, and it's really a cornerstone of a healthy soil system. The appreciation for it is definitely gaining ground, and I think we can all expect that there will be new ways to account for and to encourage it that will benefit both agriculture and the environment into the future.